It's a pleasure to be here. Today I'm going to talk to you a little bit about our science, a little bit about why we do what we do, and hopefully introduce you to a new multiplexing technology that we've really embraced in our laboratory called CYTOF mass cytometry at the end. So a brief overview is I'm going to tell you a little bit about our research aims, and then I'm going to kind of go over the same information that was already presented about Luminex assays and some of the positive and negative, mostly positive. But I'm actually a developer. I've developed over probably 100 to 200 different immunoassays and related assays using Luminex technology. And I'm going to point out some of the variables that you need to look at if you are going to develop your own assays. And then I'm going to end with some examples of how we've used the technology for multi-dimensional approaches, mostly for immuno-phenotyping complex immune responses. So as already mentioned, our lab is really focused on trauma. It is a huge problem in this country and worldwide. It's a very complex response that involves multiple immune cell types, so it's perfect for multiplexing and multi-dimensional research. We have both a mouse and a human research program, and we study not only just straight trauma from auto accidents and explosions, but we also study radiation and radiation-combined injuries for nuclear threats. Secondly, the other thing that we do is we try to develop treatments based on the information that we gain from our research. We use this information to come up with hypotheses, and we use the technology specifically to test different immunotherapies. I'm going to touch a little bit on one of our most exciting therapies that we've developed. So why do we study trauma? The reason we study trauma is it is the leading cause of death in people that are very young and productive. Oftentimes we hear about cures for cancer and cardiovascular disease, but trauma is definitely an important problem that really needs to be addressed. And not only that, after you're injured, you're also more susceptible to infections. And you can see septicemia is also in the top 10 list of causes of death in this country. So what I like about studying trauma is that it's not really a new scientific question. This is Ilya Mechnikov, and he actually studied injury. That's how he won the Nobel Prize in identifying macrophages and their ability to engulf bacteria. And what was really impressive about this individual is he did all of his studies using a microscope, an animal model, which was starfish larvae, and also visual observations and probably a pencil and pen. But today we have these great tools that allow us to study in detail multiple cell types and mediators involved in an immune response. And I often wonder what he would be doing today if he had access to these technologies, probably the same thing that we're all doing, trying to figure out what cells are involved and what mediators are involved in a particular pathological situation. So increased knowledge, I think this is pretty obvious, increased knowledge requires increased data. When I first started studying immunology, there were 2 cytokines. I often don't think of myself as being that old, but the field has advanced very quickly. There was only IL-1 and IL-2. Now, as you can see from this figure, there's a lot of different cytokines that interact with a lot of different cell types. So it's important to not be one-dimensional and start thinking about things in a systems type of an approach. The old school single-mediator assays are really obsolete, in my opinion. Multiplexing is much more in tune with the current scientific climate, mostly because it's very difficult to interpret findings and actually publish results based on one mediator these days. Good examples are the inflammatory response and T-cell mediated immune regulation. There's a lot of pro-inflammatory cytokines that people study to try to understand what's going on in a pathological situation. And T cells, as you know, have been divided into multiple subsets based on the type of cytokines they make. Also, restrictions on research funding requires much more an increase in efficiency and data collection strategies for your research. Also, we need to gather as much information as possible from expensive mouse models and also from the limited amount of human samples that you get in from clinical trials. So you all know the history of how we measure things. It starts with an ELISA, very basic assay, single-mediator assay. Then along came the flow cytometric bead assay, which you could use a flow cytometer. A lot of people use that in the immunology field. I quickly actually moved to Luminex technology because of arguments with the companies that were making the flow cytometric beads. They would not allow us to custom build our own assays. So once I found out about Luminex, we really got into the technology. And Tim Beddett, who's many of you may know, was very supportive of getting us going in the technology. So why do multiplex assays enhance your research efficiency and program? Well, as we all know, ELISAs require large sample volumes. In the day when we were doing ELISAs, we used to have to use 50 microliters of our particular biological fluid to do an assay. So this was a limitation with ELISAs, but obviously it requires more samples to generate the data. More reagents are needed to coat the wells for an ELISA. Also, you need to do multiple wells if there's more than one analyte, and that's really difficult. We used to try to measure one cytokine, take the supernatant out, and measure another cytokine, and it never worked very well. Also, if your assay doesn't work, you lose very valuable material. So at one meeting, similar type of meeting, I made the comment that ELISAs are for losers. And if you're not necessarily a loser as a person, but you're basically losing everything if you're doing an ELISA. You really need to think about multiplexing and being more efficient with your samples and with your reagents and your research budget. As we all know, Luminex assays, we do them typically with 20 microliters. If you dilute your samples, you're using much less of the sample. We make our own assays, so we can buy antibodies and couple our beads. And the antibodies, the upfront cost is expensive, but those antibodies last for years. So it's a very, very cost-effective way to perform some really great immunological type studies. As I mentioned, we developed the Luminex platform for our in-house assays. And there were a couple reasons why. As we all know, a lot of the early kits were not the best. We had ELISAs or cytometric beads to compare to, and where we would detect something in those assays, we couldn't find anything in Luminex. So I was very frustrated after just spending $50,000 on the instrument and not having reliable assays. Also, some kits detected only recombinant cytokines and not the natural cytokine. So we started to really think about building our own assays, not only because we wanted flexibility, but also because we wanted to get good data. And of course, as I mentioned, this is really a budget-saving technology. So the principles, you all know this, it was all gone through very, very clearly. The nice thing about the Luminex technology is it's built on the basics of immunoassays, so there's nothing new there. The specificity is very high as long as your antibody pairs work well and have high specificity. Some of the things that I wanted to point out, which since we are developers, we look at problems based on constants and variables. So first of all, as we all know, the beads are constant. They're very high quality beads with a specific signature in them. Your streptavidin is also a constant, but your variables are your antibodies and also your standard. So whenever we build assays, we order in our reagents and we start comparing and contrasting. Sometimes we have to order 2 or 3 different antibodies to find the right pair to work. Once we go through and build these assays, we go through all the same things that a company would for validating the assays. It was brought up for quality control, what people use. We actually take cells like human PBMCs or mouse PBMCs or spleen cells. We would stimulate them with various different immune stimuli, and we would make sure that all the assays would detect natural cytokine as well as not just the recombinant standards. Examples of how you can use multiplex technology: you can microsize your experiments. I'm actually thinking about ideas for maybe using a hanging drop technology so that we can save on cells, or where you get the cells in a very, very tight and small volume so you can get really good concentration. We've also thought about and actually used methods where we actually put the beads in with the cells to stimulate the cells, and the cytokines actually will stick to the beads right in culture. And that really increases your sensitivity a lot. And because they're magnetic, all you have to do is wash away the cells and use a detergent, and you have an instant assay. It's great for testing ex vivo cell stimulation conditions. I'm going to give you a little bit of an example of that. Again, more efficient. And we've also done some work with some people where we use very small volumes. We published a paper with an individual who was looking at dry eye syndrome and pathology in the eye, and we used tears to measure cytokines, and that was a very nice study. We've used it for organ extracts. We've used it basically whenever you can take a piece of tissue or fluid. We've used Luminex technology to detect cytokines and other mediators. Okay, so now I'm just going to give you some of my philosophy and some of the things that we do in the laboratory and how we've incorporated Luminex technology and also the CYTOF technology to use what I refer to as systems immunology, which is now being used as a term, but early on, during my first grants, I used the word systems immunology, and they had no idea what that was. They gave me a hard time, said I was descriptive, but science is descriptive. But anyways, I shouldn't complain about the NIH. It's most of the reviewers. So we approach these systems immunology or systems biology is perfect for complex scenarios such as trauma. The complexity of the cellular immune response to trauma warrants using this data-driven type technology or ideas, and it's referred to as an inductive research approach. We've all heard that you can't do good science without a hypothesis. Well, we kind of turned that around. We would ask a big question, we would generate data, and then we would develop our hypothesis. So I'm going to give you some examples of that. Trauma is very interesting. It's a very natural response that our body has to the injury. So that's it's very specific. So we have to kind of step back and look at the uniqueness of what we study, and that's why we use a systems approach. So here's some of our some experiments that we do. We have a mouse model for injury. Our mouse model is a 25% total body surface area burn injury. Now that sounds terrible when I say that we burn mice, but in fact it is the most humane traumatic injury model available because we actually kill the nerves, and the mice are anesthetized, of course, before we injure them. But this type of injury actually mimics a lot of what happens in humans, and the way we know that is because if we infect these mice in the lung, where they would develop pneumonia with a very somewhat innocuous type of bacteria, which we call opportunistic pathogens, Pseudomonas and also Streptococcus, we get a very different type of a survival response, very much like what you would see in a human. So I'm going to just show you some examples here. On the left, we have an experiment where we did a sham and a burn injury on groups of mice. We infected them at 1 day after the injury. And you can see the sham mice survive very well with the open circles. The closed circles are the animals that were injured with infection. And you can see the mortality is very high. Same thing on the right, using this gram-negative bacteria, we see a very dramatic difference and significant difference between a sham injury and a traumatic injury. So this is a big question. I mean, this is a big question that everyone wants to know. Why is it that when you're injured that you're more susceptible to infection and will actually succumb to infection? So we've done a lot of work, and I'm going to just briefly touch on some of the most interesting stuff that we found. we immuno-phenotype these animals after the injury, and we do a lot of time series analysis. So when you do time series analysis, looking at cells and mediators, it requires a very efficient protocols for being able to ask these questions or answer these questions. So what I'm showing you here is the results of what I call high throughput flow cytometry, where we use a bunch of different antibodies to identify the cells shown on the right. For example, CD4 T cells, CD8 T cells, gamma delta cells, neutrophils. And then we look at over time with a sham and a burn in response to Pseudomonas, to strep pneumonia infection. You can see different patterns of the cells that come into the lungs. And if you focus on the yellow cells, the neutrophils, you can see over time you get a nice increase in the number of neutrophils that enter into the lung airways, which we call the BAL. BAL stands for bronchoalveolar lavage. But on the right, you can see in the injured mice that there is an influx. It happens very early, but it's very dysfunctional. It goes up, and then it kind of plateaus and goes down. Similar things with Pseudomonas, a little different pattern. You can see the neutrophils come up very early in a normal mouse, shown on the left. But on the right, you can see the neutrophils don't even come into the lungs at 4 hours after a gram-negative infection. And then they shoot up to very high levels at day 1 and day 2 after the infection, mostly day 2. So this really tells us there's a huge imbalance in the immune response to infection. And we believe that this is something that we could hopefully translate into humans to understand how to develop immunotherapies. As a follow-up to this, just to prove that there isn't a difference between an injured and a normal, we can look at bacteria in the lung. And you can see on the left with the strep, you can see that the bacteria is much higher in an injured animal, shown on the right at day 4, 1, 2, and day 1 and 2. Same thing with the Pseudomonas. So as a scientist, we had a hypothesis. We said, okay, the neutrophils are really defective, so there must be a problem with chemokine production. So chemokines draw cells into the site of infection. So what we did is we designed an experiment where we wanted to look at chemokines and also other types of cytokines in the BAL fluid. And this is a very busy slide, but I'm going to point out to you that our hypothesis was absolutely wrong. If you look on the right, and this is a busy slide. The blue bars are the burn injured, traumatically injured animals at 4 hours, 1 day, 2 days, and 4 days. We did samples of the BAL. The gray and white are the sham. And if you just start on the right here, which shows all the major chemokines that call neutrophils, you can see that either with the Pseudomonas or the strep pneumonia infection, that there's really no difference. And in fact, the chemokines are actually higher in injured animals. But what we did discover was there was a defect in particular cytokines that are pro-inflammatory, which was really a surprise to us. In particular, interleukin-1 alpha and interleukin-1 beta were very, very low in these animals that were injured. And it's very clear if you just look on the top left, you can see the IL-1 alpha with the gray bars and the blue bars. There's a clear difference. IL-1 beta is much different with the Pseudomonas infection. And then we also made some observations right in the middle that IL-17, which is an inflammatory cytokine that is important in autoimmunity and autoimmune disease, but it actually is an anti-microbial cytokine. So we made the discovery using Luminex technology that we see a defect in IL-17. Now this led to us looking at other unique cell types in the lung that we would have never considered without using a systems approach. And that is the gamma delta cell, gamma delta T cell. And we went on to find that the gamma delta T cells are actually the defective cell that doesn't come into the lung appropriately, and that somehow messes up the neutrophil influx. So it's completely different than what we had anticipated. So in conclusion for this part, I'm just kind of presenting to you the systematic approach to research, inductive reasoning. I'm highlighting some of the multicellular things that we see different in a very complex situation of trauma an bacterial infection in the lung, and how we've used that to come up with a new hypothesis on developing the model for why injured people are more susceptible to infection, but also insights into developing immunotherapy. Now we get into the scary stuff. The scary stuff is that we have nuclear threats. When I first started working in radiation injury, I was a little concerned because I thought people wouldn't care about it. It was not something that we were worried about. And this was about 6, 7 years ago. Obviously now we have more threats. Fortunately, the Congress has a directed program to draw in people that are interested in studying injury into the radionuclear program for research. So we were very good at looking at post-injury type of treatments. So we began this program. We were funded by the NIH, and we used the same philosophies that we had with our trauma program to try to figure out how to boost the immune system after radiation. Now, as you all know, radiation can cause a very dramatic reduction in your cells, but in hematopoietic system. So it's very challenging. But at the same time, we were working in trauma, and we found that there was a particular pathway that we could activate through toll-like receptor 9 using molecules called CpG-DNA, which are danger molecules. And that somehow would boost the immune system to the point where we could actually restore antimicrobial immunity in trauma. So these CpG is very interesting. It's not just one type of CpG DNA. There's a number of them. So we went and screened a bunch of different CpG responses. One of the ways we did that was to perform Luminex screening early on, where we wanted to look at the difference between the class A, class B, and class C. And you can see very clearly that these different CpG molecules, when used as immunotherapeutic or as a stimulant, they produced very, very different types of responses. In particular, the class A molecules were very good at inducing IL-12, which we all know is a very beneficial cytokine, interferon gamma, totally enhances the cell-mediated immunity, whereas other types of CpGs, like the class B, were very strong at inducing IL-10, which makes them very counter-inflammatory. So this very simple experiment really kind of told us that we were on the right track using a class A molecule. So what we did is we did the same thing with injury. Except we irradiated these mice at different doses, and we set up the model a very similar way, where we infected with Pseudomonas, although we waited till 7 days, because that's really when you're much more susceptible to infection in radiation. And I think it's very clear in this slide, if you look at animals that were not exposed, which are the diamonds, you can see that there's no mortality when you infect these animals, but as you increase the radiation doses, you get very high mortality. At 6 gray, even using lower doses of bacteria, we had a very, very bad death rate. So we began testing all the variables using this drug. The CpG-2336 is the sequence we settled on, class A CpG. And you can see on the top graph, if we give the treatment at day 1, we get a complete survival response. We've also done tests where we give them at day 3 or 5, and we can see very, very good survival. So what we wanted to do is we wanted to translate this work in the mice to humans. And one way that we approached this was to think about using systems biology. We used Luminex to look at cytokine production in response to CpG stimulation by PBMCs. And then we also use the CYTOF mass cytometry to try to identify which cells are being activated by the CpG in both mice and man. And we actually used mouse blood prepared exactly as human blood is prepared. So here's the results of one of our screening assays, where we were looking at some other potential CpG sequences. And I use this as an example because it shows us 2 things. Number 1 is you can use Luminex technology to basically screen for reactivity in the immune system. So these are PBMCs where we stimulated with CpG. At 48 hours, we simply used our 18-plex assay and we looked for cytokine, different cytokines shown on the y-axis. On the bottom, on the x-axis, are different sequences of CpG. These are actually from mitochondrial DNA. The CpG-2336 is shown. That's our CpG-ODN that we use for therapy. And then we also put in LPS, which we heard, endotoxin causes endotoxemia. So we wanted to use that as our toxic type of effect. And we could match the different sequences to the positive therapeutic. Okay, so I think I'm just about done in time, but I want to introduce you to some other multiplexing ways to do research. Instead of using beads, we use cells. And what we've done is we've used the same idea using CYTOF, where we label antibodies with upwards of 40 different metals. And what this does is it allows you to resolve signals at a very high resolution as compared to flow cytometry. We built panels. We went and identified differences between mouse and man and the response to CpG. We actually looked at 170 different markers and came up with these very unique panels that are specific to responses with CpG. And I think that I've been cut off, right? Okay, oh, it wasn't working. That was a good trick, though. I like that. This is fully multidimensional single-cell analysis. And when you do this type of work, you actually have to have really good tools for visualizing your data. So what we do is we do these experiments. We then take all the data, which is like flow cytometry data, and then we allow an algorithm called VISNI or TISNI, which is like principal component analysis, to cluster cells into different sections. So on the top, you can see the cell subset markers that we use to identify cells, T cells, B cells, and monocytes. And then we have our unique markers that we look at, and wherever it looks hot, wherever it's green and red, you can see that's where the markers are turned on. So for example, CD69 is increased on the B cells in response to 2336, and also 1826, which is the class B. And we know the class B is probably not what we want to use, although it induces a stronger response. So I'm going to go through, and I just skipped that because that's a summary of what we've done. But here's the human response. So we did the same thing, exact thing, and we could actually match the different CpG sequences to what we see in the mouse. So we've actually gone from a situation where we had no idea whether this might work in a human to a situation where we at least understand that human cells can be activated in the same way by this particular drug. Okay, so just to summarize what I've presented to you, whenever we do our research, I'm always thinking about systems. I'm always thinking about multiplexing for cost saving. I believe it actually makes science more honest because you have to use your data to come up with your hypothesis. You're not just saying, oh, I want this to happen, let's make it happen. That causes some big problems. And we've been able to tackle very big questions with this technology. Both Luminex, CyTOF, and now RNA-seq really provides these opportunities. This is people who work in my lab, and obviously can't do this without funding. If you want to know more about CyTOF and what we do, we actually have a website. And we run the CYTOF resource and core for the Harvard Medical Area. If you are doing CyTOF, we can actually make antibodies for you. And you can have access to our whole collection, which is about 600 antibodies, both mouse and human at this point. And I'll stop there.